So this is your handout for protein metabolism. Protein metabolism includes protein digestion and absorption, amino acid utilization, transamination and oxidation deamination, the urea cycle, which was given on a separate lecture, amino acid carbon skeletons, amino acid biosynthesis, hemoglobin catabolism, interrelationship among metabolic pathways, and lastly, B vitamins and protein metabolism. So on your past discussion regarding urea cycle, it was emphasized about yung nitrogen balance. So in this handout, it will expand your knowledge on amino acid utilization. Okay, so nitrogen balance, it is the amount of nitrogen taken into the human body as protein equals the amount of nitrogen excreted from the body waste in as urine or as feces. Okay, so two types of nitrogen imbalance can occur in the human body. First, we have the negative nitrogen balance, which results in tissue wasting. So yung degradation, this exceeds yung protein synthesis. For positive nitrogen imbalance, the rate of protein synthesis or yung anabolism is more than the protein degradation, which is catabolism. So the results in large amounts of tissue synthesis. Okay. Next, we have protein digestion and absorption. So in protein digestion, we have pepsinogen and HCl. Okay, so the pH for gastric acidity is ranging from 1.5 to 2, and it kills most of the bacteria. Also, pepsinogen, inactive siya, pero it is activated to pepsin, and enzymes pepsin hydrolyzes about 10% peptide bonds. So large polypeptide chains pass through the small intestine, and the passage of acidified protein promotes secretion of secretin hormone, which stimulates first bicarbonate production, and second, promotes secretion of pancreatic digestive enzymes. Sa ating enzymes, we have emphasized that in the pancreatic digestive enzymes, meron tayong trypsin, chymotrypsin, and carboxypeptidase in their active forms. So protein digestion in the intestine happens also, and take note that trypsin, chymotrypsin, and carboxypeptidase in pancreatic juice release into the small intestine, and it helps in hydrolyzing proteins to smaller peptides. Okay, so amino acids liberated are transported in the bloodstream via active transport process. So this diagram depicts the flow of how dietary protein is metabolized from your mouth. So take note that saliva has no effect on digestion. In the stomach, there are two major chemicals that acts on the denaturizing of protein and hydrolysis of polypeptide bonds. So these are HCl and pepsin. In the large intestine to the small intestine, meron tayong 4 na enzymes in their active forms that hydrolyze peptide bonds. These are trypsin, chymotrypsin, carboxypeptidase, and aminopeptidase. So these ones will already give off some of the amino acids, and these amino acids is transported to our bloodstream through active transport. So amino acid pool, it says amino acids formed through digestion process enters the amino acid pool in the body. What's this? It is the total supply for free amino acids available for use in the human body. So the amino acid pool is derived from 3 sources. Of course, yung mga kinakain natin, the form of dietary protein. Second is protein turnover. So protein turnover meaning a repetitive process in which the body proteins are degraded and resynthesized. Thirdly, biosynthesis of amino acids in the liver. So only non-essential amino acids are synthesized. So take note of these 3 derived amino acid pool. So protein synthesis, about 75% of amino acid go in synthesis of proteins that is needed continuous replacement of old tissue. So replacement of old tissue, ibig sabihin, protein turnover, and to build new tissue, it's the growth. Synthesis of non-protein nitrogen-containing compounds. So synthesis of purines and pyrimidines for nucleic acid synthesis. Synthesis of heme. Ano ang heme ulit? That is directly related to hemoglobin, neurotransmitters, and hormones. So thirdly, synthesis of non-essential amino acids. And lastly, production of energy. So take note of these 4. It is used, these are the 4 different ways on how we use the amino acid pool in our body. Okay, so how do we utilize this exactly? We have here the presented degradation pathways. The degradation of an amino acid takes place in 2 stages. First, the removal of the amino group and the degradation of the remaining carbon skeleton. Kasi sa ating amino acid, meron siyang amino group, may carboxyl group, and there is a remaining carbon skeleton. Okay, the amino nitrogen atom is removed and converted to ammonium ion, which ultimately secreted from the body as urea. Okay, secondly, the remaining carbon skeleton is then converted to pyruvate, acetyl-CoA, or a citric acid cycle intermediate, depending on its makeup, with the resulting energy production of energy storage. Okay. So nakikita nyo dito sa my figure that amino acid goes to degradation in 2 portions. We have nitrogen portion and carbon portion. The nitrogen portion can be eliminated via urea. Second, biosynthesis of non-essential amino acids. And thirdly, biosynthesis of non-protein, nitrogen-containing compounds. As for the carbon portion, merong 4. Triacyl glycerols via fatty acid biosynthesis. Second is glucose by gluconeogenesis. Thirdly is ATP via citric acid cycle. And fourth, ketone bodies via ketogenesis. Okay. So we have here transamination and oxidative deamination. So what we have here is the removal of amino group in 2-step process. Transamination and oxidative deamination. So as given in your handout, an enzyme catalyzed biochemical process in which the amino group of an alpha amino acid is transferred to an alpha keto acid. So that's transamination. Oxidative deamination is an amino acid is converted into the corresponding keto acid by the removal of amine functional group as ammonia, and the ammonia eventually goes into the urea cycle. So you can have the illustrations as your guide on how oxidative deamination happens and transamination happens. Okay. So further discussing transamination, the body can manufacture the amino acids that is needed, that it needs but does not have. in an essential part of the active site of transaminases is pyridoxal phosphate. So take note of this, it's PLP, the coenzyme form of vitamin B6. Ulit, take note that the coenzyme form of vitamin B6 is pyridoxal phosphate or PLP. Okay, so transamination and oxidative deamination. Continuing, we have oxidative deamination. It's defined as a catabolic reaction whereby the alpha amino group of an amino acid is removed to form alpha keto acid and ammonia. So ano yung products? It's alpha ketoglutarate and ammonia. Alpha ketoglutarate is an alpha keto acid. So what happens during the process? Kailangan ni alpha glutamate ng isang mole ng water and in turn give off energy in the form of NADH2, or we call it NADH at my H+. Okay, this occurs primarily in the liver and the kidneys through the activity of the enzyme amino acid oxidase. Two amino acids, serine and threonine, undergo direct deamination by the hydration process rather than oxidative deamination. Okay, now we have the urea cycle. You can check the video uploaded for urea cycle as your supplementary video material. Okay, so what happens here? The ammonium ion produced by oxidative deamination is a toxic substance. So it is quickly converted to a carboxyl phosphate and then to urea via the urea cycle in mammals. Along this, you can see that the equation needs 2 ATP. Dalawang ATP, so ATP is a form of energy, right? And in the product, we have carboxyl phosphate. Plus, of course, meron pa rin tayong mafo-form na ADP. So, mas mababang form ng energy sa ating katawan. In the conversion of ammonia to urea, 3 different amino acids are involved: arginine, citrulline, ornithine. So, these ones, usually hindi natin nababanggit dun sa essential and non-essential amino acids, but take note of these 3. So the pathway for this is called urea cycle. In other books, they called it Krebs ornithine cycle. So the blood picks up the urea from the liver and carries it to the kidneys where it is excreted in the urine. Kaya ang ating urine is containing urea. Okay. So urea is a principal end product of protein metabolism and contains a large percentage of the total nitrogen excreted by the body. The urea cycle is the only means of the body para tanggalin tong urea. So any failure sa kahit anong part of the cycle will lead to an accumulation of ammonia with severe retardation. So that would be depicting a lot of problems in the body. So, malfunction of the kidneys, the liver, etc. Okay. So, we have here carboxyl group transfer. Carboxyl group of carboxyl phosphate is transferred to ornithine to form citrulline. Sabi natin kanina, si ornithine at citrulline are forms of proteins or amino acid. Stage 2, citrulline aspartate condensation. Ditong part na to, kailangan niya ng ATP. It utilizes ATP. Stage 3, argininosuccinate cleavage. So, argininosuccinate is cleaved to arginine and fumarate by the enzyme. Anong tawag? Pag meron tayong cleavage, it's always lyase. And what's the name? Argininosuccinate. Stage 4, hydrolysis of urea from arginine. So, dito sa ating illustration, we have number 1, transfer. Okay. The second will be condensation process. The third is the cleavage. And the fourth is the hydrolysis. So, tingnan nyo, pag transfer, ibig sabihin the transport of ornithine and citrulline. Okay. Excuse me. So, the linkage between the urea and citric acid cycles. Ano nga ba ang linkage natin sa dalawang iyon? Fumarate from the urea cycle enters the citric acid cycle, and aspartate produced from oxaloacetate of the citric acid cycle enters the urea cycle. So, si citric acid cycle at si urea cycle comes hand in hand, wherein the oxaloacetate goes to aspartate, papasok siya sa may part kung saan nababreakdown ang argininosuccinate. Okay. From that, the products will go to the formation of fumarate to malate. So this one happens simultaneously. Okay. Each of 20 amino acid carbon skeleton undergo a different degradation process. So 7 ang degradation products ng amino acids na ito. Tandaan nyo na ang tinutukoy natin dito is the carbon skeleton na part, hindi yung amino part. So, ano tong mga 7 products na to? It's pyruvate, acetyl-CoA, acetoacetyl-CoA, alpha-ketoglutarate, or sometimes it's alpha depicted by the Greek letter alpha, succinyl-CoA, fumarate, and oxaloacetate. So, last 4, yung 4 na nahuli, are intermediates in the citric acid cycle. The amino acids converted to citric acid cycle intermediates can serve as glucose precursors. Anong tawag doon? They are called glucogenic amino acids. Okay, so the amino acids converted to acetyl-CoA or acetoacetyl-CoA can serve as precursors for fatty acid and or ketone body synthesis. Okay, so pwedeng precursors for glucose or pwede siyang precursors for fatty acid. Pag precursors for glucose, tinatawag silang glucogenic amino acid. If it is precursors of fatty acid, tinatawag silang ketogenic amino acid. So ketogenic amino acids are amino acid that has carbon-containing degradation product that can be used to produce ketone bodies. So amino acid carbon skeleton. Paano nga ba siya nade-degrade? Okay. So meron tayo dito yung citric acid cycle. Let's start from oxaloacetate. What happens here? Kailangan niya ng aspargine or aspartase, which give off glucose. Kailangan niya din ng pyruvate. This pyruvate comes from alanine, glycine, cysteine, serine, threonine, and tryptophan. Okay. So, dito sa process na ito, meron coinciding cycle kung saan yung pyruvate, it is converted to acetyl-CoA, na yung acetyl-CoA na ito ang kailangan to produce alpha-ketoglutarate from oxaloacetate. Okay, balik tayo kay acetyl-CoA. Si acetyl-CoA, saan siya nanggagaling? It's from isoleucine, leucine, and tryptophan. So, acetyl-CoA coexist with acetyl-CoA, kung saan yung acetyl-CoA is from leucine, lysine, phenylalanine, tyrosine, and tryptophan. Also, yung acetyl-CoA na ito ay kailangan din in forming ketone bodies. Okay, let's go back to the cycle, citric acid cycle. From oxaloacetate to alpha-ketoglutarate, kailangan niya ng acetyl-CoA. Tama? And alpha-ketoglutarate to succinyl-CoA, and succinyl-CoA to fumarate, and back to oxaloacetate. So, only 2 amino acids are purely ketogenic. Ano yung dalawang yon? It's leucine and lysine. 9 amino acids are both glucogenic and ketogenic. Ano yung mga yon? Those degraded to pyruvate as well as tyrosine, phenylalanine, isoleucine, which have 2 degradation products. Okay, so the remaining 9 amino acids are purely glucogenic. So in summary, we have here the starting materials for the biosynthesis of 11 non-essential amino acids. Yan. So you can go through this. Just have to familiarize and no need to memorize. Just familiarize. Okay. So we have here the differentiation of essential and non-essential amino acids, and also kung ano ang precursors nila. Precursors nila means kailangan nila for this to be formed. So example, pag meron tayong cysteine, ano ang precursor? It's the serine. If we need glutamic acid, anong precursors niya is alpha-ketoglutarate. Okay, and so on. So we have also here hemoglobin catabolism. Red blood cells, usually we call this RBCs, tama, for the previous discussion. These are highly specialized cells whose primary function is to deliver oxygen to cells and remove carbon dioxide from body tissues. So we have this hemoglobin. It is a conjugated protein with 2 parts. Sabi natin, ang hemoglobin is globin in form, so spherical in form, and consisting of a protein portion, which we call the globin. Meron din tayong prosthetic group, which is called the heme, which contains the iron and the oxygen. Okay, so iron atom interacts with oxygen, forming a reversible complex, kung saan reversible in a sense that it can take oxygen and it can also release the oxygen. So oxygen can come on and out with it. So mature red blood cells have no nucleus or DNA. It's filled with red pigment hemoglobin. So red blood cells are formed in the bone marrow, and approximately 200 billion new red blood cells are formed daily. The lifespan of a red blood cell is about 4 months. Okay. But there are recent advancement wherein the hemoglobin and red blood cells are stored for a longer time. So hemoglobin catabolism, degradation of hemoglobin. So the globin protein is converted to amino acid and are put in amino acid pool. Sabi natin kanina, meron tayong function of amino acid pool and meron tayong mga further processes that needs the amino acid pool. So relate it to our first discussion dun sa first few slides. So iron atom becomes part of ferritin, an iron storage protein that saves the iron for use in biosynthesis of new hemoglobin molecules. So the heme tetrapyrrole is degraded to bile pigments and eliminated in feces of urine. So we have already discussed this, that bile pigments have this biliverdin, green color, bilirubin, reddish-orange in color. So these two are new now. Stercobilin, brownish, and urobilin means yellow in color. It's present in urine. So si urobilin, it gives the characteristic of yellow color to urine. Okay. So sabi rin natin, we have this term jaundice. It results from liver, spleen, and gallbladder malfunction. Results in higher than normal bilirubin levels in the blood and gives the skin and white of the eye yellow tint. So hindi lang sa ihi, napupunta rin siya sa bloodstream and even manifesting to our skin and to our eyes. So interrelationship of this metabolic pathways. So tignan nyo. Okay. So interrelationship of glucose, fatty acid, amino acid, and yung energy required. Meron tayo ditong component to be broken down. Meron tayong end products of digestion or catabolism, and yung fate. Fate meaning it's either converted to an essential biochemical, or it is nagawa siyang energy. Okay. So you can go through that, but we will not dwell more on this. You just have to be familiarized. Okay. Same with this illustration. It's just depicting that kailangan nyo ng dietary lipids. Kailangan nyo ng carbohydrates, dietary proteins. So from these three, you can form fatty acids, glycerol, glucose, amino acids. From this phase, na-form ang different na energy and essential biochemicals through lipogenesis for fatty acids, glycolysis for glycerol and glucose, gluconeogenesis naman kung pyruvate to glucose, transamination or deamination from amino acids, and the two most important cycles sa ating katawan, it's the citric acid cycle kung saan sa oxaloacetate na-form ang aspartate which is needed doon sa urea cycle in the form of ammonium ion or aspartate. Next, doon sa ating citric acid cycle, we have an electron transport chain gaining as energy. Doon naman sa urea cycle, may degradation which in turns give off urea and is excreted in the body. So these are the interrelationship of the metabolic pathways of the different biomolecules that we discussed from midterms to finals. Okay. So lastly, we have this B vitamins and protein metabolism. Structurally modified B vitamins function as coenzymes in protein metabolism as well. So 8 B vitamins participate in various pathways of the protein metabolism. So niacin, dun sa may NAD+ and NADH, also in oxidative deamination reaction. We have PLP in transamination reaction, and all 8 B vitamins in degradation and biosynthesis of amino acids. So as you can see in this figure, meron tayong 20 amino acids. Okay, so dun sa 20 na amino acid, meron tayong amino group, meron tayong carbon skeleton group. Dun sa amino group, ang metabolism niya is through glutamate. Then we undergo this to oxidative deamination where NAD+ is needed. Okay? Converting it to ammonium ion. Yung ammonium ion na yun ang gagamitin dun sa carbamoyl phosphate and it will enter the urea cycle. For carbon skeleton catabolism, meron tayong formation ng non-citric acid cycle intermediates, and these ones can go either sa citric acid cycle or non-citric acid cycle. So that's all. I hope you do good in your exams.